Welcome again to the last lesson of Christmas break. Hope you've all been enjoying your break. So today is Friday, so we have one more day until New Year's. So really the last lesson of the year. Today we're going to learn about what plasmids are and how they help us in genetic engineering. The sound again is annoying. So, for the next week and a half or so, we're going to be focusing on biotechnology, or how we can manipulate genes or use genes for our own usage. One of the things we're going to be discussing for the next, immediately right now, is how we can genetically engineer organisms to express genes that they don't normally have. So if you see in this picture, this pig has been genetically engineered the cells in his nose to glow. We've also genetically engineered rice to have more vitamin C in it. And we've genetically engineered bacteria to produce insulin. And then we also use, what we're going to talk about last, is how we use DNA to catch criminals. But today's focus is on plasmids and how we can use plasmids and get them into bacteria and get them to express genes that we want the plasmids to express. And there we have the little mousey glowing in the dark. So today, we're going to focus on how we can manipulate DNA. We'll look at a set of tools that biotechnologists work with to manipulate DNA and get DNA into, in this case, bacteria, to have them express the gene that they want and be able to get that bacteria to make that protein, purify that protein, and then use it, in the case of insulin, to give diabetics. We've talked a little bit about this. Wrong side. We talked a little bit about this idea of transformation, when we were learning about the experiment with the mice, where they put heat-killed, make sure I get this correct, heat-killed S with R, and then, and S was the bacteria that was able to kill and R wasn't. And what they saw is that something from the heat-killed S went into the R bacteria, which normally couldn't kill, to make it be able to, transform or kill the mice, to transform their protein coat, which then allowed them to kill the mouse. The word we use to describe this process is transformation. The bacteria were able to pick up naked DNA, put it into their chromosome, and as a result they were transformed, or underwent transformation, because they were expressing new genes. Whenever organisms express new genes from a different organism or an organism of themselves, this is called recombination. One thing that bacteria have evolved are these small supplemental circles of DNA that are called plasmids, in addition to their regular sort of chromosome that they have. So here we sort of have the regular bacteria chromosome that's a circle, and then some bacteria have these smaller rings of DNA called plasmids. They carry about 2 to 30 genes each, and they also generally carry the genes for antibiotic resistance, which prevents them from being killed by antibiotics. One thing that bacteria can do with plasmids is that they can exchange them. If you remember in the last video, we talked about conjugation, when the bacteria release grow a sex pilus, and then the plasmids can be replicated and exchanged between bacteria by traveling through the sex pilus. We can also, we're going to do this in the experiment this week, take a plasmid from the environment and get a bacteria to take it in through a process called heat shock, which we're going to be doing to the plasmids and bacteria. So the question is, how can we get a plasmid to have a gene in it and then get it into bacteria? In general, there are sort of 3 steps that have to occur. First, you need to cut the plasmid open. And we're going to say that these scissors here represent something called a restriction enzyme, which can cut DNA. Then you need to cut the gene that you want, or get the gene that you want from another organism. Like this could be, for example, the insulin gene from humans. And you need to cut it with the same scissor, or restriction enzyme, that you cut the plasmid with to open it. Then you need to place, and we'll see how this happens and why it happens, the gene from the organism that's been cut with the same scissor or restriction enzyme into the plasmid. At this point, this plasmid is called a recombinant plasmid, because it has genes from both the organism that you took it from, so here's the blue part, the plasmid, and then the DNA from the other organism, or in this case, as we're talking about, if it's the insulin gene, it's the insulin gene from humans. Then, once you've made many copies of this plasmid, you need to insert this plasmid into the bacteria through a process known as heat shocking. Once you successfully get the plasmid into the bacteria, the bacteria will transcribe and translate that DNA section and turn it into protein. And then if you want to get the protein out, you have to purify it through a process known as protein chromatography, get that protein away from the bacteria so that you can use it for what you want. So in summary, plasmids are used to insert new genes into bacteria. We're going to look at more in detail how this is done. So first of all, if you remember we were talking about the scissor, and biologists discovered something called restriction enzymes, okay? Or they're also known as restriction endonucleases. And they were discovered in bacteria. And they've evolved, actually, bacteria have them to protect them from viruses. Because when viruses infect them, they inject their DNA into the bacterial cell. And viruses evolved these enzymes so they could cut up the viral DNA before it got incorporated into their chromosome and make more baby viruses and cause the cell to explode. So they are able to recognize specific base sequences and cut at those sequences. So they were able to cut at only sequences that are found in viral DNA and not found in bacterial DNA, so they didn't cut up their own chromosome. The idea that they cut at specific sequences is very important for their use. They just don't cut the DNA willy-nilly anywhere. They cut it at a specific sequence in a specific way. Now, talking about those specific sequences, if you look at these silly sentences, what's in common about these experiences is that the word is spelled the same, front ways and back ways, or front looking at it in the front, looking at it way and backwards. These are called palindromic sentences, things that are the same front. If you read them from the front, and if you read them in the reverse, they give the same sentence or the same word. Similarly, enzymes recognize specific palindromic sequences, and they cut at that sequence. So if you take a look at an example, again, the scissors represent restriction enzymes. They recognize this sequence here. And when they see that sequence, they cut in this jagged pattern. Because they cut in this specific pattern, every single time they see the sequence in the DNA, they leave something called sticky ends. So if you take a look here, this is this area here. If you pull the blue one away from the other one, there would be the sticky end or overlap on this one. And if you pull this one away, there would be this sticky end or overlap. But notice, when you put them together like they did here, they're able to bind the sticky ends. So this is a sticky end here, and this is a sticky end here. They're able to bind because the sequences are complementary to each other. So just another example of sort of what sticky ends are. Let me get the pen out. Pointer options. The pen. So if we take a look, we have this sequence here that the restriction enzyme recognizes. When it recognizes that segment, it cuts in a specific pattern that leaves a sticky end here and a sticky end here. Any sequence of DNA that's been cut with the same restriction enzyme will have this sticky end, and it will be able to anneal or stick to any other DNA that's been cut with the same restriction enzyme that leaves the same sticky end. So in this example here, the blue is the gene that you want. Like, let's say this is the insulin gene, so this could be the insulin gene over here. And let's say this is the plasmid DNA. If you cut the gene that you want and the chromosome or the plasmid with the same restriction enzymes, you notice that they have the same overhangs or sticky ends, same sticky ends here, and then the same sticky ends here. As a result, when you put the gene that you cut with this restriction enzyme, the same one, and the plasmid that you cut with the same restriction enzyme, you get ends that overlap. Here we have AATT, and here we have TTAA, that they overlap and will be able to stick to each other because the sequences or the sticky ends are complementary to each other. This way, you can very easily insert this gene into the plasmid if you cut them with the same restriction ends, thus leaving the same or complementary sticky ends. Once you've cut the gene and inserted it into the plasmid, so here the yellow part is the human insulin gene, and the purple part is the DNA of the plasmid. Once you've inserted them into each other because they have overlapping sticky ends, you will insert that plasmid into the bacteria, and the bacteria will take that gene and turn it into protein, and then you'll be able to purify that protein from the bacteria, and then take the human insulin, put it in a bottle, and allow humans to inject themselves with it for their treatment of diabetes. This is only possible because the DNA code is universal. In every single organism, if you have the codon UUU in your mRNA, it codes for the amino acid phenylalanine. The code is the same in all organisms. All organisms read the code the same way. As a result, if you put a gene from any organism into bacteria, it will read it the same way and produce the same protein. So one of the things the AP exam likes is they like to say that because of the fact that all organisms use the same DNA as evidence that we all came from one common ancestor. Once the bacteria has taken in the plasmid, and this plasmid, since it has a gene from two different organisms and it is called recombinant, transformation has occurred. And if you grow that bacteria, it will reproduce the bacteria itself will reproduce and make many, many copies of the plasmid. That process is called cloning. You're sort of cloning the plasmid, or making many, many copies of the plasmid, because the bacteria are reproducing and replicating the plasmid and giving it to other bacteria as they divide. And you'll get many, many copies of that inserted gene, and then many of the bacteria, thousands, millions of the bacteria, because bacteria reproduce really, really fast, will start producing that new protein. And then as a result, you've transformed the phenotype of the bacteria, and you can use this new protein if you purify it from the bacteria for whatever you want. So just to overview the process, and we're going to look more at the details on Tuesday after school, the first thing you have to do is cut the plasmid that you want to insert into a bacteria with a restriction enzyme that leaves sticky ends. There's a lot of work to find the right enzyme, because the enzyme that you pick, and there are over like 30 or 40 restriction enzymes that cut at different sequences. You need to be able to cut the plasmid and the gene with the same restriction enzyme. And remember, the reason why that is if you cut them with different ones, they won't have the same sticky ends. Now, on Tuesday in class, we're going to look at an example of this, where we're going to look at where various restriction enzymes cut, and based on that, which restriction enzyme that we should pick to cut both the plasmid and the gene that you want to insert into the bacteria with. We're going to look at sort of all the requirements of where plasmids need to cut and not cut. Once you've done that, because of the sticky ends, here and here, the plasmid will combine with the gene, because the sticky ends are complementary, thus producing a recombinant plasmid. You will then put the plasmid and bacteria in the same tube and do something known as heat shocking, which we're going to talk about probably Wednesday in class, and then grow the bacteria. We'll see there are ways to find out which bacteria have the plasmid and which one don't. That we're also going to look at. And then once you've grown the bacteria and they start producing the protein made by this gene, you then need to purify the protein using a process which we're not going to go into, known as protein chromatography. And then that protein can be used for whatever you need it to be used for. So this video is going to run out of time, and I have 3 slides left, unfortunately. So I'm going to upload this video and then do the last 3 slides, which will only take probably about another 3 minutes, but I'm going to run out of time, and this is a good place to stop. So I'll see you in open the new video, and I'll see you in a couple seconds.